with our last talk in this amazing Restoring Reef Functions session. We've got Emily Esplandiu from University of Miami. Take it away.
Hi everyone, I'm a research associate at the University of Miami, and today I'm going to be talking to you guys about a small but important part of the research that I did for my master's thesis and that I actually am still currently working on for the next year. So up until now, coral restoration research has focused on population recovery with coral colony metrics such as survivorship and growth as the main metrics to evaluate coral restoration success. And so this is really important for species recovery, but we need a more complex approach in order to evaluate the ecological impacts and benefits that coral restoration can have on the reef ecosystem as a whole, shifting small scale to ecological scale in terms of coral restoration. So these are all the objectives that I looked at in my study. I paired both coral colony metrics that are listed here, like the survivorship and growth of some of our tagged out plants, the predation of those, coral cover, and abundance of juvenile corals. But the main focus of my study are these 4 ecosystem level metrics. So we looked at fish herbivory, coral and epifaunal recruitment, and then reef carbonate budgets, which we had the wonderful talk earlier about carbonate budget surveys, so talk about that. In my study, we had 7 different sites within Miami-Dade County, and we had 3 main treatment types. So we had wild thickets of natural staghorn coral, or Cropper cervicornis. Then we had our restored reef sites, which have 2 to 3 year staghorn outplants growing at them. And then we had control reefs. And within these 3 treatment types, we also had treatments separated between whether or not staghorn was present at those sites or not present. So we had within the thickets, thicket plots and thicket controls and so on, restored and unrestored. And so today, I only have a little bit of time to talk to you, so I can't do all of these, but I'm going to focus on the reef carbonate budgets that we looked at because it was really cool that I got to compare carbonate production at actual restored reefs compared to both wild thickets and those that are degraded. And so first I just kind of want to give you an idea of what the coral cover looked at all these different types of sites. As expected, we had the highest coral cover in the thicket sites, but we did see a significantly higher coral cover in the restored than we saw in all 3 of the control. And this is all coral, so there were some massives at the control sites, but I would say almost 100% of the coral cover in the thicket and restored is made up of staghorn coral. And so again, this is just a picture to show you an idea of what these different plots might look like. And so this is reflected here when we look at the results from our carbonate budget surveys. And for these, we did use the reef budget methodology, which was talked about earlier. You conduct detailed surveys of calcifiers and bioerodiers on the reef, and then you're able to get a net carbonate production rate from this. And so again, as we can see, it's split up between the 5 different plot types that I had. And we saw that all 3 of these controlled, so the unrestored and the restoration sites, the thicket control, and the control reef themselves all had negative carbonate production. So all of these sites are in a state of net bioerosion, while both the thicket and restored sites were in positive net carbonate production, so positive accretion. And this is telling us that the high density staghorn aggregations that we're creating as a result of coral restoration are promoting net carbonate production. And so as we can kind of see here, even though the restored sites are not as high of a level as the thicket sites, they're moving in that direction, whereas the control sites are in a state of net bioerosion. So this is also telling us that these plots where we have restored staghorn coral, the staghorn is outweighing the bioerosion. So the bioerosion is still there at all of these sites, mainly parrotfish, but we can see that the staghorn that we're outplanting there might be or is outweighing the bioerosion impacts on these reefs. We also saw that coral cover had a significant positive relationship with carbonate production. And as we can see, the coral cover in the restored plots here, so we have percent coral cover and then net carbonate production, is moving in the same direction as, or in a positive direction towards the coral cover of the thicket. So we could suggest from this that in the future carbonate production, as that coral cover increases, will increase towards the similar values of the thickets as well. And also in a study by Perry et al. In 2013, they found that a reef can shift to a state of net carbonate production when coral cover increases beyond 10%. I found in my study that the threshold was 2.78%. So this could be due to Acropora cervicornis species-specific contribution towards carbonate production. You know, they're a fast-growing branching species, so this is kind of what we expect. But this also might mean that we don't have to outplant as many in a specific area at a site in order to combat that bioerosion at the site. I have a lot of other metrics that I looked at in my study, so please, if you're interested, come find me later. We'll be here all evening at the poster, so I'd love to talk more about these. And I also am doing another year of new sites for my study, so looking forward to that. But as a whole, my study is serving as a framework to shift coral restoration from a small scale to ecological scale so that we can better understand the reef resilience of the ecosystem. And by pairing these colony level metrics with ecosystem level metrics, we can gain a better idea of how the success of these corals that we're outplanting is impacting other components of the entire reef ecosystem. And I just want to thank my lab and my funding and Ian Enoch's lab at NOAA. They helped me with the carbonate budget surveys and modifying it. And if anyone has any questions, that's it.